So here we're starting the conversation about the immune system. And I usually break it down into sort of three levels of protection. One of them is what I call intrinsic immunity, like intrinsic barriers such as mucus or cilia. Okay, so you should know where they can be found and what's the point. So mucus is a trapping device. It also contains a bunch of antimicrobial chemicals. Cilia, especially cilia in respiratory tract, they move mucus upwards. So this is called a mucociliary elevator. What happens to the mucus that the cilia lifts up to your larynx?
It moves it to your throat so you can swallow it.
Yeah, you swallow it. And then acid in the stomach kills it. We have low pH. So low pH, well, obviously stomach and vagina. Vaginal pH can be altered after antibiotic treatment, so that often promotes the unwanted microorganisms to grow. Skin is a little bit acidic. Now, sebum that sebaceous glands produce, the oily secretion, contains fatty acids that seem to be antibacterial. And then this pervasive protein, lysozyme, that is found in pretty much any secretion from tears and sweat to mucosal secretions, non-specifically kills bacterial cells. Dermicidins, special proteins produced on the skin, also kill microorganisms. And last but not least, microbiome provides a pretty good barrier from the invasion. Now we're done with the surface barriers. Let's jump to the innate immunity and discuss cells that comprise the innate barriers. All white blood cells can be divided into two groups: granulocytes and agranulocytes. So the main characteristic of granulocytes is the presence of these cytoplasmic granules that contain various compounds, inflammatory compounds, toxic compounds, and so on and so forth. So there are three granulocytes that are found in circulation: neutrophils, eosinophils, and basophils. So again, highlight, all of them are found in circulation. Functionally, neutrophils, which gain the name neutrophil because they can be equally well stained by acidic and basic dyes, target bacteria. Whenever you see increased number of neutrophils in patients' CBC, it's pretty much a signal that somewhere there is a bacterial infection. How do they kill them? Well, one way is to phagocytose bacteria. Another is to release chemicals of respiratory burst, pretty much hydrogen peroxide. They also can increase inflammation and produce very cool response called neutrophil extracellular traps. When a neutrophil dies, and it is a kamikaze cell, when it dies, it releases its DNA. And that DNA wraps around bacteria and prevents it from spreading. Now, eosinophils target, for the most part, eukaryotic pathogens such as fungi or protozoa. Now, you can imagine that due to the size limitations, eosinophils can't really phagocytose, so they have to rely on other ways to get rid of microbes. They also release chemicals that incur cellular toxicity in the pathogens, chemicals of respiratory burst, and they can increase inflammation. Unwanted role of eosinophils is in allergy. It's been shown that in patients with, say, allergic asthma, the number of eosinophils in the walls of trachea is considerably higher than in the patients with no asthma, just a regular infection. Basophils. Well, the term allergy, I want to scratch it out actually. I want to say just inflammation. So basophils, these guys, contain intracellular granules, and inside those granules you can find chemicals such as histamine, bradykinin, okay? Things that produce inflammatory response. Now, when basophils migrate to tissues and become resident there, they turn into mast cells. Mast cells in the tissues are not solely, but largely responsible for your inflammatory reactions. Now, if you ask yourself, for me, question, but why do we have mast cells that, like bad? They probably have some other role, and we still don't exactly know what their role in the immune response is. Now let's jump to agranulocytes. So, 3 cell types that are found in different compartments: monocytes, macrophages, and natural killer cells. So let's start with the monocytes. Monocytes are found in the circulation, largest white blood cells, and they can do phagocytosis, but their main role is to develop into macrophages. Do you follow? The trigger for that development is inflammation. Once macrophages have been produced, once monocytes developed into macrophages migrate in the tissues and start destroying debris, viruses, bacteria, infected cells. So they kill it through phagocytosis, and they also increase local inflammatory response. Now the third type of cells, which are agranulocytes, natural killer cells. So this image number 3 here shows you the NK cells. Now, why do we call them natural killer, and why are they in the innate response? Innate response does not really distinguish between individual microorganisms. So your innate immune system will respond to some bacteria, viruses, different viruses in sort of the same fashion. Am I making sense right now? You with me? Okay. Now, NK cell is very similar to adaptive immune cells that are called T cells. It's similar because it kills using the same approach. So check this out. This right here is a healthy cell. NK cell has a receptor. Okay? One of the names for that kind of receptor, and I'm not making shit up, is don't kill me signal. So healthy cells normally, no seriously, healthy cells normally produce molecules that tell NK cells don't kill me. Now an NK cell finds, don't kill me and kill me signals. Okay? When NK cell finds a kill me signal on a cell, well, and it kills it. The mechanism, apoptosis. if you're curious about kill me, don't kill me, Nobel Prize in Physiology and Medicine for checkpoint inhibitors, cancer treatment. Okay? Now, so we figured the cells. How do we recruit them? Okay? So, this is the infection right here. Okay? So this infection, obviously, is the injury to the tissue, and the injured cell release chemical signals, these molecules called cytokines. And the name cytokine, cyto, cell, kine, movement, okay, they make cells moving. So these cytokines get in the bloodstream. Would you agree with me? And eventually the blood brings those cytokines where? One word starts with E. It's a tongue-in-cheek question. In the human body, where does blood go? Everywhere. So these cytokines reach everywhere. Importantly, they reach bone marrow and stimulate leukocytosis. Yes. So now we have increased level of white blood cells. Make sense? So these white blood cells get in the bloodstream, and now they have to determine where the infection is. So you see these leukocytes emigrating to the site of infection. That happens due to two things. First, inflammation increases the expression of adhesion molecules, and they serve as like a Velcro for white blood cells. And inflammation also increases vascular permeability, so for cells it's easier to crawl up. Here's what happens. Your white blood cell, garden variety white blood cell, will attach, roll, adhere, extravasate. I don't think that the verb to extravasate is in the Merriam-Webster, but we're going to use it anyway. The term extravasation means out of the vessel, extravasation. So those cells that do this, mostly neutrophils and monocytes, once they are in the tissues, they follow these inflammatory chemicals, just like Hansel and Gretel followed the trail of breadcrumbs, which is called positive chemotaxis. So monocytes become macrophages, and these two guys destroy pathogens. Now, both of them can phagocytose. Neutrophils can also release DNA in the form of NATs, respiratory burst, that kind of stuff. Also, think about the increased vascular permeability. It means that more plasma gets from the blood vessels into the tissue and washes away debris. The last but not least, plasma contains clotting factors. Tissue damage will activate clotting factors, and it will result in the formation of insoluble fibrin. And this fibrin will trap microbes. Okay, we don't have a lot left, but this is an extremely important thing here. So, I'm thinking where to start. Remember, in the first unit, we were talking about gram-positive versus gram-negative organisms, right? And we mentioned that gram-positive organisms, they have teichoic acid. For example, in the cell wall, right? Now, gram-negative microorganisms, for instance, have lipopolysaccharide. So we can say that lipopolysaccharide is a molecular signature of gram-negative organisms. And teichoic acid is a molecular signature of gram-positive. So these molecular signatures that are associated with a pathogen are called pathogen-associated molecular patterns. Okay, so, for instance, some bacteria have flagellum. One of the key proteins, flagellum. Pattern, pattern. Some viruses, many viruses actually, have RNA genomes. Having an RNA genome, pattern, pattern. So I want you to process that this pathogen-associated molecular patterns, or PAMPs. So imagine this is a pathogen, and this is the pattern. Let me make it a little larger, otherwise this is pathogen-associated molecular pattern. Am I making sense? So these patterns are recognized by your immune system. So that's your immune system, and this is pathogen pattern recognition receptor, PRR. Does that make sense or not? I'm not going to move on until I get the confirmation from everybody.
So to restate it, the pattern recognition receptor is just a receptor that's found on
our cells.
On our cells.
Yes.
Cool.
And it recognizes those pathogen-associated molecular patterns.
I see. Okay.
Like a molecular pattern would be, okay, you can recognize a spider. I bet you can, right? Because you know how they generally look, 8 legs, all that kind of stuff. So there are some attributes to a spider that are common to all the spiders and allow you to recognize a spider. Pathogen-associated molecular patterns are molecules that are common to a certain class of microorganisms and allow your cells to recognize that type and respond appropriately. Okay, is that clear? Full disclaimer, there are a lot of them. I mean, PRRs, the receptors, there are a lot of them. I give you just a taste of it. These are so-called toll-like receptors, one type of them. Okay, and they are membrane-bound receptors. Obviously, please do not memorize any numbers here. What I want to illustrate is that receptors can be found in the cell membrane. And they would recognize things like bacteria, for instance. Or they can be found in the membrane of the endosome, inside the cell. 3, 4 out of 10 question. Which type of microbes will be mostly recognized by the receptors that are sitting in the endosomal membrane inside the cell? Say again? Any virus. Because the virus is intracellular. So the virus is taken into the cell by the endosome, so you have a virus here. Okay? This is the transport, basically. And this is where you want receptors that can recognize it. Okay? Now, there are receptors that exist in the cytoplasm. My point is, they're numerous. I just want to lighten up because we have like a couple more things to talk about. The history behind the named toll-like receptor. So it means that receptor is like a toll, right? It's not related to toll roads, though. The first receptors of that type were identified in Drosophila, the fruit fly. And the scientist who identified them, allegedly, when he saw the results of the definitive experiment, he was German. He exclaimed, like he saw the results, he was very excited. He said, toll, which in German is like cool. And they named those receptors in Drosophila toll receptors, so literally cool receptors. And they were discovered in humans, in mammals. Scientists didn't have anything better than to call them just toll-like, which shows you the absolute lack of creativity in scientific nomenclature. Okay? Now, when phagocytosis happens, what's going on? Super simple. Look, there's a bacteria that's being engulfed, and now it sits in the phagosome. Phagosome fuses with the lysosome, which contains a bunch of digestive enzymes, forming phagolysosome. Those digestive enzymes and acid and reactive oxygen species destroy bacteria, and then waste is released. Making sense? The process is really simple. Thing to remember, two main phagocytic cells, neutrophils. These are kamikazes. They phagocytose and die. Macrophages can phagocytose multiple times. Clear? Complement or worst nightmare of any immunology student. Okay, so complement is the system of proteins. Okay, it's produced in the liver, not associated with any specific cells. One of the oldest system of immune protection in animals. Okay, so complement has to be activated. It's a cascade. Does that make sense? Like a domino effect. So hear me out. What you need to know: 3 activation stimuli, the convergence point, and the effects. Let's start with the 3 stimuli. Classical pathway. Everybody took ANP2, I presume. Yeah, okay, so then you're familiar with that. So imagine you have a pathogen, and you have an antibody bound to the pathogen. Okay, you may remember that this part of the antibody is called FC. FC, just FC. Okay? FC fragment. So that FC fragment activates complement. So you see that antibody, it's not just like some random roaming antibody. It has to be bound to the surface of the pathogen. Clear? It's complex. Second route is lectin pathway. Okay? So imagine you have a pathogen. And on the surface, it has a sugar called mannose. That sugar can bind another sugar. that is present on human cells called lectin. Okay, so there's binding. And when that happens, you have activation of the complement via lectin pathway. And to prove my point about the lack of creativity, the third pathway is alternative. It means that there is some stuff on the surface of a pathogen that will activate complement. Which stuff? There are options. Does that make sense? Please do not waste your time on memorizing all the intermediate steps. I will never ask this. I want you to notice that all three pathways basically converge on the same molecule of C3b. It's a convertase. Let's explore what are the responses. So first response, first effect of complement activation is increased inflammation and phagocyte recruitment. Okay? Second option, one of my favorites, I always do, is the formation of so-called membrane attack complex here. So a bunch of other complement proteins get assembled like a Voltron together, forming a pore. Every time I use Voltron cultural reference, I feel how much older and older I become. Okay? So they form a pore, and that disrupts the distribution of ions, action potential, and eventually a cell shuts down. Okay? So far, inflammation, membrane attack complex. What about opsonization? So imagine you have a pathogen. Okay? And you have complement protein C3b that sticks to the surface of that pathogen. Okay? So if a macrophage has to select between the pathogen in its native form without C3b and the pathogen that is surrounded by C3b, that will be the choice for the macrophage. These green circles represent opsonins, and the entire process is called opsonization. The easy way to remember, let's say you're buying a hot dog, a bun and a frank. Are you going to add something to it? Yeah? I mean, I'm not asking what, you're probably going to ask mustard or ketchup or relish or combination, right? Because without it, it's pretty bland. So this is the hot dog without any condiments. You can eat it, but it's not really appealing for macrophage. When you add condiments, which are opsonins, that's when macrophage really gets on. Okay? Go ahead.
It's just like a marker for the macrophage?
Pretty much, yes. And guess how macrophage, this is 4 out of 10 question. Guess how macrophage recognizes it? Binds to it. Using what on its surface? Close, but don't be that specific. What is the last letter in PR stands for? Macrophages have complement receptors that will recognize opsonins and bind to them very specifically. Does that make sense? So you see that it's pretty motley crew, let's put it this way. The last bit that we're going to talk about is interferons. So there are two types. These are proteins. Okay, let's highlight the important points. Okay, proteins. They are proteins, okay? So when pattern recognition receptors on the infected cell or in the infected cell, when they recognize the pathogen via recognition of PAMPs, that recognition triggers the pathway that often results in the expression of the interferon gene and production of interferons. There are two types, 1 and 2, and they are produced when there is an infection. Does that make sense? Not every infection results in interferon production, but infection is required for interferon production. So these interferons, since they proteins, react with the interferon receptors on the surface and lead to the expression of so-called interferon-stimulated genes, or ISGs. ISGs, they function, limit, or stop pathogen replication. Okay? Does that make sense? So, first, I want you to appreciate in this scheme that you see you have an infected cell here. Okay? It sends out interferons to other cells to warn them that shit just got real, you know? They don't do anything to a pathogen. It's still, hey, we have a problem, mount up the response. And they can signal in three main ways. Actually, they can signal on the same cell that produced them. So the cell gets infected, releases interferons, interferons bind to its receptors, send it to apoptosis. They can migrate to adjacent cell and work with it, or they can be released in the blood and act in the endocrine. So this is autocrine, this is paracrine, this is endocrine fashion, and act in the distant cells. Okay, make sense? They are used to treat some cancers. They were used to treat Hep C. So now they are used to treat some severe infections like, let's say, hemorrhagic fevers. Here's the problem with interferons. Imagine you have a zombie apocalypse, okay? And a bunch of zombies start storming Tri-C campus, okay? And I get beaten by zombies, right? And I run in here and tell you, hey folks, there's a bunch of zombies, get ready. That makes sense. Now imagine the different situation. Zombies are already on campus, and they're killing you all. And somebody calls and say, by the way, there are zombies. Way too late. My point is, interferon therapy works before or very, very early in infection, like super early. It works the best. If you start giving it late, we did some studies on yellow fever. If you wait as much as 12 hours, it wasn't a human infection, it was model. But if you're like 12 hours, effects are minimal. If you treat it like 3 days before, that's great, but it's really hard to predict the future. Okay, so the efficiency is pretty low and side effects horrible. Okay, so we're going to continue our conversation, finish up the immune system next time. So we're actually going to focus on the mechanisms of adaptive immunity. And first I want to highlight the difference between the adaptive and the innate immunity. So innate immunity, as you may remember from our last week conversation, is not suited for a very specific pathogen. So it kind of offers broadly protective responses, but it doesn't differentiate between, let's say, influenza virus and respiratory syncytial virus. Adaptive responses are specific, and they're targeted against a very specific pathogen. Because of that, they require the exposure. So without you being exposed, for instance, I bet none of you have any immune memory about, say, Marburg virus, because you've never been exposed to it, okay? Because of that exposure and because of multi-step activation process, it is slower than innate, but more effective, because the response is systemic. But perhaps the most important feature of adaptive immunity is that it can form what is called immunological memory, which means the immune response that is produced after the second exposure is faster and more robust. So let's take a look at the components of the innate immunity, adaptive immunity. So there are two main types, cellular and humoral. The Greek word humor means fluid. So humoral immunity is mediated by B cells, which after the exposure to the pathogens become plasma cells, which in turn produce antibodies. Antibodies are released into the circulating blood. Blood is fluid, therefore the response is humoral, mediated by soluble proteins. T cells, after the exposure to an antigen, a pathogen, form a plethora of different, what we call effector cells, which can kill infected cells, help other immune cells to become more efficient, or in different ways regulate the immune response. Both B and T cells are lymphocytes, small cells with the large nucleus, and all of them, just like any other cell of blood lineage, originate in the bone marrow. The main difference between the two is that in order to become functional, both B and T cells have to mature. B cells mature in the bone marrow, their birthplace, while T cells move to thymus and mature in thymus. Once B and T cells underwent maturation, they migrate into the lymphoid tissue, more specifically lymph nodes, where they are waiting for their exposure to a pathogen. In order to make this conversation smooth, let's agree on something. I'm going to stop my annoying habit of asking you if it makes sense, and if it doesn't, you let me know immediately. Just interrupt me. Deal? Like seriously. So what exactly those immune cells respond to? They respond to an antigen. What is the characteristic of an antigen? It is large and complex molecule that is importantly foreign, and I'll explain what it means, for your immune system. Usually, since our conversations are concerned, they rotate around humans and mammals in general. Foreign means different species. So large and complex molecules, the most common antigens are proteins, but it can be carbohydrates, can be DNA, and so on. So they all are large and complex. Now, some examples of the molecules that are large and complex but aren't foreign. All people in this room belong to the species Homo sapiens. unless I don't know something about some of you. If I will collect your blood and isolate the protein albumin from your blood and inject it into my blood, I will mount no immune response because we belong to the same species. So albumin, your albumin, does not serve as an antigen for my immune system. Okay? Now, if I collect human albumin and inject it into, say, a rabbit, for a rabbit, human albumin is foreign, and rabbit immune system will mount a response against it. So it will serve as an antigen. My point is that you shouldn't treat antigens as something that is non-human. It's something that is foreign for the species we're talking about. Good? Now, molecules that are complex but aren't large enough. You can think of fairly large organic compounds, say cholesterol, steroids, synthetic steroids, for instance. They're not large enough. So they're complex, but they can't serve as an antigen because immune system, its quote-unquote resolution isn't enough to quote-unquote notice those things. Now, what about something that is large but too simple? I think Mona has a plastic water bottle, right? It's probably polyethylene, polymer. Polyethylene, the molecules of it are enormous, but they are extremely simple. So this is why, generally speaking, plastics are not recognized by your immune response. On one hand, it's convenient because you can make hypoallergenic stuff. On the other hand, we're full of microplastic, which our immune system completely ignores because it's not complex enough for it to recognize. Is that good? Okay. Now, here's what's interesting. So this is your garden variety antigen, obviously, schematic. Now, your immune system does not recognize the entire antigen, the entire protein, or the entire carbohydrate. Just like you, when you think about some person that you know, you aren't thinking, oh yeah, I remember them. They have 2 arms, 2 legs, they had, those are not features. Features are, I don't know, curly hair, small nose, long hands, something like that, okay? So those specific features that actually trigger the immune response are called epitopes or antigenic determinants. Now, you can see that this generic schematic antigen carries 3 generic antigenic determinants. What if the antigen molecule would be bigger? Would it have more, potentially, antigenic determinants? Yes. It's not the universal rule, but by and large, the larger the protein, for example, the more antigenic determinants it contains. Partially, it can explain very marginal superiority of Moderna COVID vaccine over Pfizer COVID vaccine. Pfizer vaccine contains a fragment of the spike protein, while Moderna vaccine contains the entire spike protein. Now, the difference in efficacy, there is some, but it's not the difference that we would consider to be physiologically or immunologically significant. It may be statistically significant, but from biological standpoint, it's not. Okay? When we're talking about the differences between like 95 and 97%, come on. So what exactly recognizes the antigenic determinants? It's recognized by the antibody. Antibodies are proteins that consist of two light and two heavy chains that are connected by disulfide bonds. If you look at this molecule, does it look like a fork? Yeah, it kind of does. So this part, the handle, is called Fc region. It's pretty much constant. It doesn't change from antibody to antibody. In the term Fc, F stands for fragment, and C can be interpreted as cell. Your immune cells can recognize and bind Fc region. The prongs of the fork are variable and termed FAB region, which stands for antigen binding fragment. Now, you can say that there are two prongs. Each prong can bind an antigen. So how many antigens can this antibody bind? Two. I kind of confused you. This and these are antigens. So you have to have both. So two. Your reasoning about four was correct. I just did not explain that this is the entire thing, the antigen binding site. Question?
That's called the FB region?
FAB. Yeah. Fabulous. Fab region. Okay? Now, right away, to clear any confusion, if you take any antibody that is produced by your immune system, not synthesized in the lab, the antigen binding sites, two of them, both of them, recognize the same antigen. Moreover, they recognize the same antigenic determinant. Okay, so look at this image. This is the antigen binding site, and this is the antigen binding site. Both of them recognize this green antigenic determinant. Now, can you synthesize sort of a tricky recombinant antibody that would have two different antigen binding sites? You can, but we're talking about what happens not in the lab, but in the body. So now we're going to talk about B cells. Let's start with B cell development. As we mentioned, B cells are produced and mature in the bone marrow. And on their surface, B cells have the B cell receptor. Also known as immunoglobulin D. B cells are among the most numerous in your body. I mean, I discount red blood cells because these guys, you cannot topple them in terms of the numbers. But there are a lot of B cells, and there are a lot of different populations of B cells that carry receptors with different antigen binding sites. All right? So, these receptors, these immunoglobulin D molecules that are embedded into the cell membrane of B cells, will be able to recognize, they are pre-synthesized, a wide variety of antigens, but each B cell has its own unique specificity. When B cells during the B cell maturation, B cells are tested for a very important feature called self-reactivity. And we call this negative selection. Humans produce a fair number of different proteins and carbohydrates and other potential antigens. Right? Now, do you want any of your B cells to mount an immune response against your own antigens? So I usually say that the B cells are asked a question. If they are activated by self-antigens, that happens during maturation. There are two possible answers. One answer is yes. You don't need those cells. You don't need self-reactive cells, right? So self-reactive cells they die in the controlled cell death known as apoptosis. If the answer is no, they survive. So ideally, as a result of that negative selection, you have a huge number of different B cells which antigen binding sites on the T cell receptors can recognize a huge variety of antigens. Now, let's say, I'm going to make a scheme again. You have three populations of B cells. Population 1, population 2, and population 3. The picture that I'm about to draw is not physiologically correct, but I'm trying to make a point. So let's say the population 1 has one specificity of B cell receptor. Population 2 has another, and population 3 has another. So here comes the antigen. This antigen, which receptor is it going to bind? Number 1. And that selects that population of B cells called the clone. So these B cells become chosen, and they go through proliferation. What does that mean, proliferation? Yeah, a lot of them. And also differentiation. What does that mean, differentiation? Not quite different types, but, well, you can say that, yeah, they become more specialized. And two populations that emerge, one of them, I'm going to put the letter P, that's plasma cell. Now, these plasma cells will produce a bunch of antibodies that will have the same specificity as the B cell receptor. Another population will be memory B cells. They will remember the pathogen, and next time you get exposed, your immune response is going to be much faster and much more robust. The process of proliferation and differentiation of a specific B cell clone is called clonal expansion. I always find it easier to draw it rather than use a pre-prepared images. However, I would not discount these images. Here's what I want to highlight. Check this out. Activation of B cells can occur by them simply binding, recognizing the antigen without any help from another category of adaptive cells, T cells. Can T cells help? Yes. Will it make the humoral response better? Also yes. But conceptually, B cells can recognize and mount a response to an antigen without the participation of T cells. Now, how many antigens do you think exist in the world? Different antigens? Well, probably billions. Think about how many different organisms there are. Bacteria, viruses, I'm not even talking about like mammals or plants or whatever. So the question is, how do we create that practically infinite array of different B cell receptors, different antibodies with different specificities? It's inconceivable that humans have billions and billions of different genes. If anyone took ANP1 relatively recently, in a year, give or take. No? That's sad. Do you maybe remember the mechanism of the smell sensation? Yeah, but the mechanism, accordion mechanism, no? When you need a variety in nature, you work through combinations. Just like a license plate on your car contains 3 letters and 4 numbers, but there are 26 letters in the English alphabet, and there are 10 possible digits for every position out of 4, it makes pretty much unlimited number of license plates. In a similar way, the variety of those antibodies is ensured via VDJ recombination. So, turns out that the variable, the constant regions of antibodies are formed by the combination of several gene fragments in the process that is called VDJ recombination. Let me illustrate it for you. I'm going to blow it up. So we're going to number the gene segments here. Obviously, I will never ask you to memorize like how many V segments we have. It's absurd. So that's maybe too thick. Okay. 1, 2, 3, 4, 5, 6, 7. 1, and that's going to be 8. 1, it's going to be 4, So you pick 1. Let's say you pick number 2 out of your V segments. And then let's say you pick number 5 out of your D segments. And then you pick number 2 out of your J segments. So you have that combination, 252, which makes the B cell receptor specific to an antigen. That's it. If you pick another combination, the structure of the FAB fragment will be different, and it will be specific to a different antigen. the total number of combinations is approximately 300 billion. Obviously, you need to remove ones that can be self-reactive. It's going to be, okay, 25, 30,000 maybe. Nothing, drop in a bucket compared to 300 billion. So please understand what is going on in the VDJ recombination and be able to sort of, if I give you different answers, what's going on, you pick something like gene fragments are combined, ensuring the variety of antigen binding sites. Not for the exam, but for your own education. Does the development of B cells stop there? Turns out it doesn't. So now, think of it this way. You've got your B cell. It was exposed to the antigen, and now it is a plasma cell that produces antibodies. Okay, now B cells, the memory B cells, we're also going to do memory B cell. Memory B cells undergo a process, very interesting process, called somatic hypermutation. First of all, juxtapose the terms somatic and germinal. What are the differences? Somatic cells versus germinal cells. Huh? No. What is germination? How do you understand the term germination? Huh? Reproduction, yeah. Germ cells are reproductive cells. So effectively, oocytes and spermatocytes. That's it. That's a germline. Somatic hypermutation means that these mutations occur in B cells but are not passed down to the offspring. This somatic hypermutation further increases the variability of the FAB fragments, allowing your immune system to sharpen, to hone your immune response even better. COVID vaccine provides me ample examples for immune responses. If you get single COVID shot, are you protected against dying from COVID? You are. Not 80-90%, but you're protected. Okay? Now, 4 weeks later, you get a second shot. What about now? Much better, because that second shot stimulates, boosts somatic hypermutation. And then you get your third shot. Those memory B cells maybe decided to kind of, retire, but you don't let them because you boost them again, and then they'll go further somatic hypermutation. Now, it can't go forever. At some point, the precision is pretty high. And there was a guy in Germany who received more than 3 COVID shots. Anyone who wants to venture a guess how many COVID shots he received over a span of 2 years? 217. He was doing that for other people who didn't want to get COVID shots, so he posed as some other person and received the vaccine record and gave it to them for money. Turns out, 217 vaccinations over 2 years, very marginally better than 3 vaccinations over 2 years. And the guy was fine other than that. He had a pretty strong immunity, but now, when antibodies are produced, well, let's start with this. There are 5 classes of antibodies. Let's knock out the boring ones. IgD, immunoglobulin D, serves as the B cell receptor. IgE participates in two responses. One, it is produced in response to various parasitic infections, and two, it's the antibody that's responsible for allergic reactions. Then we have IgA. Small amount of immunoglobulin A is produced, released into the blood, but most of it is secretory. Secretory IgA is a dimer, and it's secreted in, many fluids, tears, saliva, every mucus, and breast milk. So those dimers from breast milk, along with immunoglobulin G that is passed across placenta, I'm running a bit ahead of the train, they give babies, newborns, passive immunity. Okay? Now, IgM. I'm debating myself, should I introduce? I think I should. IgM, as you can see, is the pentamer. So you have 5 antibodies here. 1, 2, 3, 4, and 5 that are all connected. How many antigens these 5 antibodies can bind? 10. Right? It's a lot. IgM are produced first, period, first. So when your B cell starts producing IgMs, kind of in a hastily manner, are they optimally fit to the antigen? What do you think from the common sense? Probably not ideal. They good, but they're not the best. Clear? So I decided I'm going to introduce these terms, not for the exam, but I think every medical professional should know the difference. First term is affinity. How specific the binding is. So when we say the affinity is high, it means that, let's talk about antibodies. It means that antigen very quickly and readily binds to the antibody, and it's really hard to take it off. Okay? It's a very good glue. But then there's a term, avidity. I decide that I'm going to hang a picture on the wall. Okay? And I have amazing glue, just brilliant. One touch somewhere in the center will be enough. Okay? Can you imagine that scenario? Now imagine I'm still hanging that picture. I have kind of lousy glue. What should I do to make sure that it doesn't fall? A lot of glue in different points, right? IgMs have very relatively low affinity for each FAB fragment. But there are 10 FAB fragments. So it's a lousy glue, but there are multiple points of attachment. Okay? These antibodies, IgMs, have low affinity but high avidity because they're 10 sites. Another important feature of IgMs, they activate complement. Remember classic pathway that we talked about last week? They're really good at it. And then the celebrity of all antibodies, IgG. It's a majority of the antibodies, and it is the only antibody that crosses placenta. They can, but it's minor. They can be. I'm gonna tell you straight up. It's sort of, yeah, they can, not their primary role. So I don't think in our limited time it's reasonable to talk about it. So IgG, the only antibodies that can cross placenta, they're also real good at activating complement. And the Fc fragment, this one, can bind to phagocytes. That will become super important. Now, why I focus so much on placental crossing? Some of you will go into nursing, and one of your part or PA, like not some, you will interact with patients directly, and you're going to talk to them about, prophylaxis and vaccines and stuff. Currently, it is recommended that pregnant women receive vaccines such as COVID vaccine, RSV vaccine, and DTaP, diphtheria, tetanus, and acellular pertussis. The reason for it, you can't give those vaccines to a newborn child for a variety of reasons. So when a woman receives those vaccines, she mounts an immune response. And those immunoglobulins, IgG, they cross placenta, end up in the neonatal circulation, and protect babies for quite a while. Okay? So it is considered, valid, half a year. Breast feeding extends it because IgA kind of also plays the role, but transplacental immunoglobulin G, about half a year. And then babies become eligible. Probably the biggest issue in terms of pediatric infections, measles. Newborns are eligible for their first measles shot at about a year. So this is why population immunity is so important. They literally aren't, so even if mom was vaccinated, she may not give them enough immunoglobulin G to give them enough protection. Okay, and there's some details important which we can't really talk about while mothers shouldn't receive MMR vaccine. Okay, so what happens when you are exposed to the antigen for the first time? Well, it takes some time for B cells to encounter the antigen because it has to enter your lymphatic system and to differentiate into plasma and memory cells. So plasma cells start producing antibodies. The first response is always IgM. And then you see how IgM decline and IgG increase. Those are same plasma cells that produce these antibodies, but they undergo the process that is known as class switching. They switch from IgM class to IgG class. Eventually, antibody levels in your blood decrease. It is normal. It's called contraction of immunity. So now, sometime later, you receive a second exposure to the antigen. IgM is still produced, but look at the IgG response. Much more robust and last much longer. It will go down. That's normal. Remember, you always have plasma cells. This is why this IgG response, the secondary response, is so much faster and so much more robust. And then let's say we again talking about COVID vaccine, you receive a third shot, IgG spike even more, and then they contract. Do you think that these responses, spike and then contraction, and eventually it's really hard to detect antibodies in the blood, are they common for all infections? No. One of the most unique antibody responses is produced when people get vaccinated against smallpox. There are studies demonstrating that people who were vaccinated, and remember, last vaccinations against smallpox in the United States, not among military personnel, that's different, among civilian population, happened in the mid-70s. People who received the vaccine back then, antibodies are still detectable. It's one of the most long-lasting vaccines known to people. Okay.
So is the IgG response so much faster because of the memory cells? Is that right?
Yes.
Okay.
Yes. Yes, exactly. Humoral immunity can be classified based on how it is acquired and whether or not the person sees an antigen. If it's acquired naturally, so I'm talking about infection or breastfeeding, it's, natural immunity. If you need human action, vaccination or intravenous immunoglobulin, okay, that is like this. That's artificial. Now, what does that mean, exposed or not exposed to antigen? So think about this. In case of infection or vaccination, your immune system is exposed to the antigen. Whether you are sick with COVID or you receive vaccine, your immune system will mount its response against a specific antigen. In case of passive immunization, the person acquires antibodies without being exposed to the antigen, like a baby acquires antibodies from the mother. Okay? Or at the dawn of COVID pandemic, Eli Lilly was the first company that released monoclonal antibodies. Okay? And I think now, I don't remember which company it is, the drug called Pemgarda. Monoclonal antibodies. They are produced somewhere else, and you just get them infused. So overall, you should be able to combine the terms, like ask you vaccination. What type? Artificial active. Right? Okay.
Do you have equal outcomes in immunity system or?
You mean passive versus active? That's a great question. Of course not. Because antibodies themselves are proteins and they have half-life. When you receive, this is why infant immunity that the infant acquires from the mother goes away. They just get destroyed, and infant didn't see the antigen. Okay? That actually, sorry, I can't resist. I want to bring a problem for you. Okay? It's a great illustration of the importance, like how sometimes your thoughts, something that seems obvious, isn't really the answer. Are you all familiar with a disease named poliomyelitis? Yeah? So until 1953, it was a common disease among children. It's interesting, in the 19th century, it was practically unheard of. Okay, so here's the illustration. No, I'm not going to draw anything. I'm just going to tell you. Wait, it's not for the exam, but I'm super excited. I'm a kid in a candy store. Imagine end of 19th century, hygienic conditions are pretty poor, right? So flushing toilets may be a luxury. polio is transmitted by fecal-oral route. There's almost no polio. Eventually, hygienic conditions start to improve. Clean running water, toilets, flushable toilets. So fecal contamination in the environment decreases significantly. What do you think happens to polio cases? They spike. When hygiene was horrible, everybody got polio. Only 1 out of 200 cases of polio becomes paralytic. So people got sick, recovered, and carried those antibodies, and probably were boosted by those infections over and over. If it's a woman, she gives birth to a child, and the child receives maternal antibodies and becomes protected from polio for about 6 to 7 months. Just right for the time when the child starts exploring the world, crawling around, picking those fecal material, and sticking it into his or her mouth. Kids are immune at this point. So when they receive the virus, they don't get sick. But their immune system now saw the antigen, mounted a lasting response, so they grew up without polio. When we cleaned up, we removed that crawling exposure, so when kids got to 4 or 5 years of age, they're not immune, and they get polio. I'm not endorsing just living in filth, but so what?
Confusing us now.
Huh?
You're making us confused now. What to do? No, you're fine.
No, I'm saying that this is an importance of maternal antibodies and how they can play. Okay, like malaria. At what age kids start to die? 6 months. That's when maternal immunity wanes. That's when, I mean, they can die earlier, but like they really start to die at 6 months because they're not protected. What can antibodies do? They can, with the viruses, they can neutralize it. So they bind to the virus, block it from binding to the receptor. With toxins, they can precipitate them. So toxins just can't do their thing because antibodies surround them. With bacteria, they can agglutinate them, making clumps, and this makes bacteria an easy target for, say, your macrophages. They can, obviously, activate complement, and they also can opsonize. We used the term opsonization before. Remember? Complement can opsonize. So what about antibodies? Imagine this is the pathogen. And these are very simplistic antibodies. Okay? Now this part, the handle, is FC. So far, I think I'm not saying anything new. What is new is that cells called macrophages have the receptor, FC receptor, that can recognize it, and suddenly macrophages very specifically start destroying whatever pathogen is surrounded by these antibodies. So not only complement, but antibodies too can opsonize pathogens and direct them for phagocytosis. What I want to emphasize is that antibodies don't lyse or kill bacterial cells. They tag them for destruction. It's like I was talking to a friend of mine, a colleague today, and he explains immune system as mafia. So antibodies is the hit that's being put out. Like, that's your target. by itself, antibody doesn't do anything, but it gives every other immune cell the information of what to kill. This is one of my least favorite things to explain. I'll try my best. So we have, well, not only we, but mammals and other animals have a group of molecules on the surface of the cells called major histocompatibility complex. I'm going to try to briefly explain the function using some drawings. All nucleated cells, that excludes platelets and sperm cells, all nucleated cells have MHC class 1 on their surface. So I'm going to draw 3 cells. The first one is normal. The second one is infected with a virus. And the third one is cancerous. All these cells carry this MHC class 1. The function of that complex is to stick an antigen that is produced in the cell, some kind of a peptide, on the cell surface. So your normal cell will signal that it's normal by sticking out a self-antigen. And your immune cells, they not trained to deal with that, so they're just going to pass by. When a cell is infected by a virus hijacks cellular metabolism, and it's virus peptides that are expressed on the surface. Cell signals to the immune system, hey, I'm not in good shape. I should be killed. Cancerous cell, same story. I should be killed. Okay? And hopefully your immune system, specifically T cells, can find those bad actors and destroy them. Here's the problem. And there are 2 types of MHC. I'm going to get to MHC class 2. There's a problem with T cells. T cells are stupid. If T cells see an antigen, like just antigen, they're not going to respond. They're like dumb, like seriously dumb. They have to have that antigen explained or presented to them. And that's done by antigen presenting cells. There are 3 of them. Dendritic cells, macrophages, and B cells. I am going to be focused on dendritic cells. So whenever I say antigen presenting cells, further, I'm in dendritic cell. We're not going to get into the weeds of macrophages and B cells. Okay? So, MHC class 2 is found only on antigen presenting cells. So, a macrophage or a B cell, do they have a nucleus? Yes. Do they have MHC class 1 then? Yes. Do they have MHC class 2? Also yes. Antigen presenting cells carry both types of major histocompatibility complex molecules.
So if they have class 2, they must always have class 1. Is that how that works?
Class 1, you have to be nucleated. That's all. Class 2, you have to be antigen presenting cell. All antigen presenting cells are nucleated. So they have both. Your liver cell on its surface only has MHC class 1. Your neurons, MHC class 1. because they're not antigen presenting. They have not, no, they have none. That's super important. We not at the, remind me about red blood cells when we get to transplantation, or when we get to fucking red blood cells. That's also going to work, okay? Right now, the last bit that I want to mention, just to put it in your head, we're going to comment on this later. If you think about the term histocompatibility, histo refers to what? Tissue. So whether tissues are compatible. All people have different MHC molecules. This is why transplantation is the pain in the ass. Because if one of you will receive my kidney for your immune system, so if this is my kidney here, and this is somebody else's kidney, let's say they are that, also normal, but their kidney, their cells carry different peptides. So the immune system is used to pink, but it's not used to light blue. So when light blue gets into the body, the immune system goes absolutely berserk. Keep it in mind. Now, red blood cells, they don't have this shit. This is why you can take red blood cells from one person and put it into another person. Clear? And there's no rejection. And the same reason sperm cells don't have this. Because otherwise it would be a disaster. Immune system will just kill them all. So this is an overview of what happens when T cells mature. So it all starts, I'm going to enlarge it. It all starts in the bone marrow. They generate it there, and then they go to thymus. In the thymus, they mature into two distinct populations, CD4 T cells and CD8 T cells. CD4 and CD8 T cells are functionally different, and they recognize different MHC molecules. CD4 recognize MHC class 2. CD8 recognize MHC class 1. What is the product of 4 multiplied by 2? 8. What is the product of 8 multiplied by 1? I call this rule, a mnemonic rule, always 8. How do you know which one does what? There are two pairs of multiplicants, 2 and 4, okay, MHC2, CD4, or MHC1, CD8, okay? So how do they get educated? Dendritic cells present the antigen to them. Now they get educated and they become effector cells and memory cells. Now that's exclusive recognition. CD4 recognize class 2, CD8 recognize class 1 is sometimes referred to as MHC restriction. Let's step back for a second and talk about the selection of T cells, the positive selection and the negative selection. So let's think what happens. Let's think about qualities that T cells have to possess. One of the qualities is the ability to recognize MHC itself. Okay? So effectively, T cells are being asked two questions. Question number one. Can they recognize MHC? If the answer is no, they're useless. Right? They go in apoptosis. If the answer is yes, then they tested for self-reactivity. So, activated by self-antigens, question mark. If the answer is yes, that's autoimmunity. You want to kill those. So if answer is yes, apoptosis. If the answer is no, they move to circulation. Not circulation, sorry, lymphoid tissue. Does the logic make sense? I hope so. Because what if so this is, the first step is positive selection, like that step, and that step is negative selection. If your positive selection fails, what you're going to get? You're going to get dysfunctional T cells. T cells that cannot do anything, right? If your positive selection fails, what you're going to get? What kind of disorder? Autoimmune. I realize what I want to do eventually with my slides is to show you where every discovery that got Nobel Prize. Remember, we're talking about VDJ recombination. That's Nobel Prize, physiology and medicine. Mechanisms of autoimmune disease. Directly prize this year, and tangentially prize several years back. I will make comment on that as well. So, now let's focus on CD4 cells first. Not much to say, except for this CD4 T cells. Recognize MHC class 2. They become activated, and they form helper and memory helper cells, in addition to a bunch of other cells. Now let's figure out how they work. So this is your CD4 T cells, cell that's being activated. So the first pool that's formed is memory. Then there are two helper T cells. I'm not requiring you to distinguish functionally between Th1 and Th2 responses. I just want you to know that helpers, T helper cells, they can increase the activation of B cells. So sometimes B cells need a little push from CD4 T cell to produce a lot of antibodies. And they also stimulate macrophages to become more effective at destroying infected or cancerous cells. They also develop into so-called T regulatory cells. I'm ignoring Th17 because that is such a rabbit hole. I don't want to go there. Then they have Tregs, T regulatory cells. That's a Nobel Prize this year. Like literally 2 or 3 weeks they announced. So Tregs decrease the immune response. Because you don't want that immune response to go on forever, otherwise, there's going to be a problem. Okay? So the Nobel Prize this year went to 3 people. One discovered, and I don't want to present myself as a fool, so I'm probably not going to dare to explain exactly. Basically, he showed that there's a population of T cells that can control autoimmune disease. I believe the guy was working with mice that develop autoimmune conditions, and he removed a thymus from them, where maturation happens, very early in life, and they did not have autoimmunity later. Because kids have very active thymus. Okay? I may be wrong. Don't take my words to the bank. I'm going to look it up. I need to see what the combination of experiments was. And 2 others, the molecular mechanism, basically the genetics of T regulatory cells. Not only this, not only helping and dampening, CD4 T cells are also critical for the proper activation of CD8 T cell. So in summary, they make B cells better, they make macrophages better, they regulate immune response, and they make CD8 T cells better. Anyone is a fan of Taylor Swift's Boyfriends team? No? You familiar with those? They coach Andy Reid. How does it look like? Spherical. It's pretty much spherical. It's going to be faster for you to jump over than walk around. If you will put that coach on the field and ask to run 10 yards, you're going to have a coronary. Right? You need to call an ambulance right away. Take him out from the sidelines and the team is going to lose. He never plays, but without him the team fails. CD4 T cells is the coach of your immune response. It makes everybody play better, and without it, does nothing directly to infected or cancerous cells. But without it, the whole system falls apart. There's one disease that very specifically eliminates CD4 T cells. It's HIV infection. And the immune system completely falls apart. Everything else is produced, it's just not effective. Now, what about CD8 T cells? So these guys are killers. When they are activated, here, they recognize the infected cell, because that infected cell, I'm going to go back in a minute. That infected cell carries the fragment of a pathogen on its surface, carries an antigen. So they recognize this, and they can only recognize MHC class 1, and that's MHC class 1. So they bind to that cell and kill it via apoptosis, either through so-called Fas receptor, Fas ligand pathway, basically shaking hands and killing, or perforins and granzymes. Here you can see perforins form pores in the membrane of the infected cell or cancerous cell, and granzymes enter and also trigger apoptosis. Looks good. Agreed? It's a pretty efficient system. Works great. Should kill cancer. Why doesn't it? It's not a question. It's like, honestly, it's 8 out of 10 difficulty-wise, because you gotta know certain things. Cells can produce so-called checkpoint proteins, not for the exam, for your own education. These checkpoint proteins, when on the surface of a cell, communicate with these killers, with T cells, and tell them, don't kill me. Sometimes jargon for those signals, don't kill me signal. Like seriously, it's called don't kill me signal. Cancer cells produce insane number of don't kill me signals. Okay? Have you heard about so-called checkpoint inhibitors? Yeah? Opdivo and Keytruda. Antibodies that block checkpoint proteins. So cancer cells stop screaming, don't kill me. And suddenly CD8 T cells, quote unquote, see them and kill them. Steve?
Okay, but if you did that, like, wouldn't that affect all of your cells? Like if you inhibited checkpoint protein?
Yes, but the thing is, your immune system does not concern about your own cells because it's not self-reactive. You see? You can start mounting CD4 T cells like, oh shit, we have the cancer here. So basically the killing is done by your immune cells. The killing. The drugs allow your immune system to recognize the cancer. Now, it's not broad spectrum. It's been shown to work in melanoma. It's been shown to work in non-small cell lung cancer, but I believe it failed with breast cancer. And here I'm going to bring you the one, I bring you the most recent stories and discoveries. So there was a study in patients with non-small cell lung cancer who were on that inhibitor, checkpoint inhibitor. And so the study compared two groups of patients. One group was like on just inhibitor, that's it. And another group, within 100 days of the start of the therapy, I don't remember if it was before or after, and that's my fault. Basically, they got COVID shot. And they compared survival. I need to have a little app on the phone that would make a drum roll. Median 3-year survival in patients who did not receive COVID shot, 18%. Who did, 37%. Twice as much. The median survival time, no COVID shot, 20 months. COVID shot, 37 months. They prolonged the life for about twofold. And turns out, it's not about COVID, it's about mRNA. Because they tried other mRNA preparations and they had the same effect in mice. They studied different preparations in mice, but in humans the effect was absolutely clear. So it seems like the shot stimulates your immune response and reduces the number of checkpoint proteins on the surface of the cancer cells, making the therapy more effective. Okay? So, like literally one of my friends has melanoma. I'm going to tell her, if you are on these drugs, go ahead, get a COVID shot. Summary. I'm not going to reiterate everything that I said. I just want to highlight some stuff. Look, when we talk about those different branches, like innate versus adaptive and so on and so forth, you have to keep in mind that they're connected. So for instance, helper T cells help B cells, so cellular immunity helps innate. They also activate macrophages, so it's a link to innate immunity. Okay? Sorry, cellular helps humoral, cellular helps innate. Antibodies can activate complement, so it's all interconnected. That's my point. Okay? Now let me